characteristic for cells is that they have to maintain a homeostasis and an environment inside their cells which is different than outside the cells. Okay, so crucial to this understanding is plasma or cell membranes. They are semi-permeable, which means, of course, they can let some things in and other things they keep out. Okay, so that's what's meant there. All right, so here I have a crude drawing. Here's the actual model is called the fluid mosaic model. Okay. What it basically says is that it's the base structure of it is a phospholipid bilayer. All right, so you guys recall there's the polar head, so the phospholipid head there, and then the two fatty acid tails coming down. All right. Okay, so we call that amphipathic, okay, which means it's got dual qualities. It's got a hydrophilic end and it's got a hydrophobic end. All right. Okay, so if you put a bunch of phospholipids together, they naturally form what is called a bilayer. So the membrane is called a phospholipid bilayer. Okay, so this would be outside. This would be outside the cell, extracellular, and then this is inside the cell. Okay. So it forms that barrier there. So it is a bilayer. Right? So now you know from your study of lipids that they can pack together quite tightly. So interspersed within that bilayer, you have another lipid called cholesterol. Okay, so cholesterol is it's a ring structure. Here's a little model of it there. I think another one here. They're interspaced all throughout. And what that does, it kind of breaks those phospholipids up a little bit to allow them to move around each other. Okay, so that's why it's called a fluid model, because those lipids just always are moving around each other. Okay, so it's a kind of a fluid environment. Also embedded in that phospholipid bilayer are a lot of different proteins. Okay? And if you recall with proteins, the R groups can have different characteristics. Some are hydrophobic and some are hydrophilic. Okay? So if you take a protein and put it in here, the hydrophobic part of the protein would tend to go into the bilayer where you have all the hydrophobic tails, and the hydrophilic R groups would tend to go towards the outside, so they can align that way. So pretty ingenious, right? So a lot of those proteins provide some of the functions of the cell. You know, crucially, you need to get some things into the cell and some things you want to keep out. Okay, but crucial to most cells is oxygen. Okay, so you want to get oxygen in. Now remember, oxygen is O2, so it is a nonpolar molecule. Okay, because of that being nonpolar, it can make its way easily through this thick layer of nonpolar hydrophobic area. So oxygen can diffuse through easily. Same with carbon dioxide. Carbon dioxide actually is a nonpolar molecule because of its structure, and so it can leave easily. So nonpolar things can get through easily, but something like water would have trouble getting through this bilayer. Okay? So to help them, they have what are called transport proteins. Okay? Specifically for water is aquaporins. Like here's an example here. Okay? So it's a protein that has hydrophilic area up here, hydrophilic area here, and in the middle it's hydrophobic. Okay? On the outside, but on the inside it creates this hydrophilic channel. Okay? So small molecules like polar molecules like water can make its way easily through it by osmosis, going from high concentration to low concentration. Okay. There's also proteins embedded which are transport or carrier proteins, okay, which molecules like glucose can lock into on the outside, and then there'll be a conformational change in that protein, and it'll move that protein inside. Okay, so that's called. We'll talk about that a little bit later, but that's facilitated diffusion. Okay, so there's carriers which go all the way through, so those would be called transmembrane proteins. There's also proteins embedded which are called enzymes. Okay, so like a good example of that is this enzyme called ATP synthetase. It's on the inside surface of a mitochondrial membrane, and so it acts to make or synthesize ATP. But it's embedded right inside of the membrane. Okay, so there's enzymes embedded into the phospholipid bilayer to do that. There's also receptors. Okay, a thing called transduction. okay, moving things across. Okay, so hormone would be a good example there. So a hormone from the outside can lock into one of these proteins, and that causes a conformational change in that protein, and that triggers a reaction inside the cell. So the hormone does not actually have to get inside the cell. It just locks in here. Once it does that, it causes a change, and that triggers some reactions to occur inside the cells, which we'll look at that stuff a little bit more later on. That's called transduction. There's also just an identity of the cell, like type ABO blood. Okay, so there's glycoproteins on the surface, okay, which can identify that cell as being that cell. So identity there. Adhesion, cells need to adhere to things a lot of times, or to each other. Okay, so there's glycolipids to that. Lipids, okay, glycos or carbohydrates tend to be kind of sticky. Okay, so that allows adhesion to other things. Okay, so you have the carbohydrate end out here, and then inside here it's anchored with a lipid component. Okay, so adhesion. And then the cytoskeleton. Of course, you know, cells, not all cells are just round. Okay, they can be long and skinny, or they can be oval shaped, or they can be flat. Okay, but there's a lot of times there's what is called a cytoskeleton inside the cell, within the cytoplasm. Okay, but has to be anchor proteins on the inner surface of the membrane, which the cytoskeleton can then anchor onto and dictate the shape of that cell, okay, which is crucial.